conservation, the establishment of the national green bank in 1996. So when during 1984, when the first the germplasm was conserved at -18°C, so we were having some 280 accessions. But seeing the awareness and the efforts and the guidance given by our peers, we could establish our national gene bank, and which currently is having more than 4.62 lakhs of the germplasm accessions of more than 265 different species. So these are the efforts, and in this direction, I think the awareness programs like the current training program and the other programs like the field day, germplasm day, which we are holding every year. So they are very important, and particularly for the younger generation, this type of trainings, they are very essential. Generally, the younger generation, they are skipping from the germplasm activities because they maintaining and then further use, that is a very lengthy process, and generally the outcome is also not that good. But to sustain the crop improvement program, sustain our the breeding programs, the maintenance and its sustainable use, I think that is essentially required. And now the various conventions and even ITPs, FRA, so they are now more concentrating on conservation, its sustainable use, as well as the benefit sharing also, that is also coming. And with the establishment of our PPFRA, the plant breeders' rights as well as the farmers' rights are also there. So definitely we should take care of that. And because if we are properly maintaining, properly documenting and getting the germplasm registered, so definitely the benefit sharing is also one option. From that, rebreeding activities, I think that in today's context, that is the most important activity, and those who are involved in the PGR activities, so they should definitely concentrate on those activities, although the immediate outcome is always not there. But if we see our future food security issues, so definitely rebreeding is also one of the very essential activity, and our colleagues, particularly the younger generation, they should come forward and they can have some good pre-breeding program and good publication in the peer-reviewed journal. So that is also a reward in that direction, and as a long-term strategy for the food and nutritional security. The participants and number of the representation which Dr. Gautam has just mentioned. So I think that is a very diverse group, and a large number of participants. They are through the virtual mode. That is one advantage. So they are participating in that. And I was seeing just our resource persons. So I think the countries, the well-known, even from the CG system, also some of the resource persons have been taken. So all the participants, they are requested that they should, I think, interact. They should participate seriously, interact, and further use this knowledge in their day-to-day breeding program for conservation of genetic resources, as well as their use, sustainable use in the plant breeding program. So with these words, I'm sure that the type of program which has been charted out, so that will be definitely very useful for all the participants, and definitely will help in the food and nutritional security of our country, as well as for enhancing the income of our farmers. So with these words, I'm very highly thankful to Director Saab and coordinators for inviting me and giving this opportunity. And thanks to all of you for joining this program. And I'm sure that this will be really very beneficial in your day-to-day crop improvement program in your respective institutes. Thank you.
Thank you very much, sir, for your encouraging words and extending all possible support. Now is the time to listen views and thoughts of Dr. R.C. Agarwal, Chief of today's function. Sir, please.
Thank you very much. For colleague from headquarters, Dr. D.K. Adarji, Dr. G.P. Singh, director of this great institute. And I welcome Dr. G.P. Singh for this family. I am family member of this since my career began. It's not. Welcome Dr. G.P. Singh for this very important institute of ICR. I can say backbone now. This entire agricultural research, and First of all, I would like to congratulate you for organizing this important meeting, important workshop, training program, I can say. In education division, we received many training programs, proposals for many training programs. But because of paucity of funds, we selected only a few. So, and we are being one of the very important organization, and this topic being very important. So we considered that we must go ahead with this. And we know the reputation also that how in the best possible way the trainings are organized by this institute. So it's always our pleasure to consider any proposal which comes from this. I don't think there is any proposal for CAP from this institute. So the Center of Advanced Faculty Training, that also I think we must propose, we are going to revise it. So if then it becomes a regular phenomena and a regular process to train the faculties, because this PGR has to become a very important topic for discussion. You know, at the global level, whatever developments have happened in the last, I can say 2 decades, whether it's a Convention of Biological Diversity, whether it's a the TRIPS agreement, whether it's a WTO, whether it's a ITPGR, whatever it is, the base of all these things is just the PGR. And in any of these TTs, international developments, you will find that they always talk sustainable utilization of these resources, then what is the benefit sharing, and that is the common thing in all these international developments. And for that, in India, whatever developments have happened, I have seen your topics for discussion on this PPFR also, and you have many other topics connected with this policy. So whatever developments have happened in India also, for that, the NVPR is at the focal is at the focal point, because nothing, whether it's a National Biodiversity Authority or whether it's a production of plant varieties, they also have their gene bank in NVPR itself with the technical help of NVPR. So nothing can move without this. So a strong network is required through such training programs, which is already there, but I think we can further strengthen using this. Many of you attended this latest GB meeting also in Delhi, which was organized, and one of the most successful meeting of the GBs. Till now, 9 GBs have been conducted, but as per this ITPGR Secretariat, this was one of the most successful conducted in all the sense, technical and arrangements and other things. There also you see the latest developments like this digital sequence information, how it was discussed and how further they want to take it up and how the countries are divided. So let us keep through such training programs our all scientists, all PGR workers fully aware with all these international issues. As a DDG education, I'm also requesting all my vice chancellors that all these international developments should be also taught to the students. If it is not the part of the course program, at least through some lecture series, through some special seminars or conferences, the students should know irrespective of their discipline. You may be a student of economics, you may be a student of statistics, you may be a student of genetics or whatever, pathology, but at least some core subjects have to be taught. And PGR is one of those core subjects. Because for development of anything, you require ultimately the base material. And base material only can come from this. Recently, just 2 days back, I was in, I think you have heard Heartfulness Education Trust. And the name of that center is Kana Shantivanam in Hyderabad. So one aspect, of course, is meditation. It's one of the largest center of the world for the meditation. In their hall, 1 lakh person at a time can sit And there are many other things. But the other beauty of that center, why this was my fourth visit, was that the agricultural intervention, if you see at their center, and especially the conservation of the beaver. More than 200 species of the Western Ghats they have brought and they are conserved in Hyderabad in a barren land. So that was really a very good intervention. And all rare and endangered species also, they don't have much experience, but it is their passion. They want to showcase, and successfully they have developed a man-made rainforest. And how so that kind of thing we want to replicate everywhere, so that all these threatened, endangered species, rare species, they are conserved in the same habitat by creating same habitat in which they are there. Instead of, in addition to we keep in gene bank through tissue culture, through the pollens, through any other mechanism, or through ex situ. So that kind of new technique, that how we can create such environment outside their original habitat and then conserve them successfully. So that was really very appealing, and I think some of you can visit to that center, and it is getting a lot of appreciation, and because of that intervention and spirituality, of course, the president of that center has been awarded for institution recently, just 3-4 days back. So that is really, I was impressed by seeing that aspect, the conservation aspect. So many other organizations are also coming forward, and they really understand and realize the importance of such kind of interventions. Slowly, I have found, because I have been involved in this organization since it's when it was developing in 1989, and if you see the growth, I think it is in the exponential rate. Every time when you come, you find always new things. Yadusaf, I can just tell you the kind of the wheat germplasm, how it was characterized. I think it was a big record in the whole world. No other organization can take such a huge task. More than 20,000, 22,000 exercises, characterizing them by involving so many organizations and then recording data and then doing everything. It requires a lot of resources, a lot of manpower, and a lot of vision for doing all that. So that's why I'm saying that always we find some new things, and that has really brought the reputation also of this institute to the forefront. That now in such a big way, and similarly you have done for other crops also. Yeah. And documenting everything through this IT platform, again really something good, which we have achieved through these organizations. And we have already a very good network with all the institutes. But as education DDG, I can just say that whatever support is required for training programs, capacity building, we are there to help. Recently, we had a meeting with, I think all of you know, there is a new commission which has been founded by the government last year, and that is Capacity Building Commission. CDC, we call it. So that is that commission is the apex body now for any capacity building kind of thing. Every department has to have one organization, which there can be multiple organizations also. Maybe the LVPs also can be considered, who have to follow certain standards being developed by the capacity building commission. And then those centers will be the part of the CDC, and other organizations also will keep coming there once you are in the list of that CDC. So that meeting was chaired by Honorable MOS, I think just 10 days back. And now we have made the nodal center for that, Dr. Srinivas Rao. So that also will help us in identifying the areas, in identifying the standards, in identifying the linkages with other organizations, So presently, we generally focus on most of the participants, they come from NARS. Maybe from the traditional universities, like now we are going to declare as one of the center for the entire country for many topics. They have already visited the particular center. And that CBC is directly, the chairman of CBC is directly reporting to the honorable Prime Minister. So it's the growth of CBC is really again exponential. Whenever I meet to CBC, they always come with new and new developments, whatever they are. So through that also, in that meeting, I also proposed that for PGRY, we cannot consider NVPR as one of the center for as per the accredited norms. They will have some accreditation also. They will send some team to accredit. So let us move in that direction also. Let us have some standard protocols, standard operating procedures for the capacity building programs, standard modules, online modules, MOOC courses. So many more things which are required. Okay. So that if anybody who cannot come and who didn't want to have the attendance in your scheduled training programs, so offline anytime they can attend it, they can get the certificate and they can learn as per their own interests. Okay. And the new term which has been evolved after this COVID is the micro learning. Micro learning. Okay. So we have to also focus on the micro learning, so that anybody in the small capsules can know. I want to know only the utilization. Okay. I can sit for half an hour. I can just listen to your lectures. I can listen to your online resources, and then I can make up my mind. Okay, what is this utilization? Next time, after 2 days, I am interested. Okay, let me see what is the characterization. What is the other parts? Okay. So all those things we can learn. So that all those things will come under this new protocol which is coming out. But let us prepare ourselves so that more and more persons get involved. In addition to the great work which we are doing, actually collecting, actually conserving it, and then factorizing it and then utilizing it. So I want to just compliment again to all of you, and I would like to thank you once again for calling me to this important workshop, this training program. And I'm once again sorry for keeping this short, and because as Dr. Yadav has told, we have another meeting at 11:15. That meeting was scheduled by DG at 10:30, but I told that I have already scheduled this particular training program. Then he told, okay, no problem, you can come at 11:15. So I would like to just once again compliment you and thank all of my colleagues. For me, it is a homecoming. So always homecoming is very pleasant. So it's a great pleasure for me to be part of this. Thank you. Thank you.
Thank you, sir, for your kind words. Certainly, we will try to be at your expectation. As we know, in our tradition, felicitation is a practice to honor the guest as a mark of respect. So I request our director, sir, to felicitate our guest.
आपको कंडीशन भी है
हमारा हो गया सर पुराने
Thank you, sir. Now I request Dr. T.S. Suda, Nodal Officer, for vote of thanks.
Sir, thank you very much. Good morning to all. First of all, I would like to thank the respected chief guest, Dr. R.C. Agarwal, Honorable DDG Education. We are highly impressed with your inspiration thoughts, sir, regarding PGR management, and I'm happy that you will help us in sanctioning some training in the future. Last year also we have submitted, but we could not make it. Sincere thanks and gratitude to Dr. D.K. Yadav, DDG seat, for his benign presence in this function and elaborating use of emphasizing the use of PGR. Thank you, sir. And I'm also grateful to the HR unit who has helped us financially and provided the fund, because in the institute there was no fund. I'm thankful to Dr. Jain for helping us. I'm very thankful to the program director and director of the institute, beloved director Dr. G.P. Singh, who helped in every respect. Wherever paper we put up, he signed without reading it. Thank you, sir. And I am thankful to all the HODs, in charge units, Dr. Veena Gupta, professor, Dr. Ashok, principal scientist and former director. We are also thankful to Dr. Gautam, Jyoti, who are convener, and Dr. Kuldeep, coordinator, and convener Dr. Kuldeep Tripathi and co-convener Ravi, Subhas, Mamta Singh, who have framed all these program. And administration, we got every type well. We are thankful to the all administration, Pamanji, Prashantji, who has helped us in every respect. And I'm very thankful to Arun Sharma. We were free from this all logistics. His team is so active that we were not worried how it will be managed. And Ankur Tomar, Shivangi Mathur, they compiled all information from the participants. I'm thankful to all the participants and resource persons from different organizations. Thank you very much, all the persons. And special thank you, Sunil Archak, Vijay Mandal, for making this all the online program. Thank you, sir. Thank you.
ले लेंगे पहले डॉक्टर साहब को अच्छा
Now we will break for the tea.
ग्रुप फोटो नहीं लेंगे
मैंने अगर आप फोटो खिंचवा देंगे ना तो ये लोग चले जाएंगे फिर चाय ही नहीं है
बाकी चाय
अंकुर जी, अंकुर तोमर, अंकुर तोमर, मेरी आवाज सुन रही है? अंकुर तोमर, आप वीडियो बंद नहीं करेंगे वहां से, आप अपनी वीडियो बंद नहीं करेंगे। HB सिंह कमेटी रूम का वीडियो नहीं बंद करेगा कोई भी। अंकुर तोमर, मैसेज टू यू। शिवांगी, अंकुर तोमर बार बार वो HB सिंह कमेटी रूम का वीडियो बंद कर देता है। माइक यूजर अपना माइक और स्पीकर बंद नहीं करेगा कोई भी। HB सिंह कमेटी रूम के लिए है मैसेज ये।
Am I audible?
Yes, sir.
Okay. Now, I hope you all have joined.
66 have joined only.
All participants are requested to join immediately as we have to start our session from just now. Listen. वहीं बैठो तो mute करना पड़ता है।
Here,
यहाँ से
we have taken it.
सर म्यूट हो गया है आपका, म्यूट हो रखा है आपके यहाँ से।
This is requested to all participants. Please maintain your attendance on regular basis because this is required for when we are marking attendance for each and every lecture to be online. And try to, as I requested yesterday, try to switch on your video because this is required for screenshot which we are going to popularize through social media platform of our MBCGR websites and Twitter handle. And now we are going to start our this session of evaluation of participants for impact assessment, in which I request our training coordinators, Dr. R.K. Gautam and Dr. K.S. Suda. Dr. R.K. Gautam is presently heading the division of Journal Class evaluation, and he is also our training coordinator. And Dr. K.S. Suda is a nodal officer of HRD, ICR, and BPGR. So I request you both, sir, please. This is over to you.
Thank you, Kuldeep. Am I audible to all the trainees?
Yes, sir.
Okay, very good. So once again, I welcome all of you to this training program. So far, as you witnessed, the inaugural program was very nice. It was excellently organized by our team. So weather also here is quite pleasant in Delhi. And there is a saying that well begun is half done. But we are to maintain this pace throughout the training program for 21 days. We have tried to have the excellent lectures from the, excellent resource persons, not only from our country, but also from abroad. So I request all of you to take advantage of this training. And we firmly believe that training will be very useful to all of you. Now, with these words, I request my partner, Dr. K.S. Suda, he is also training coordinator, to say a few words on this occasion. Yes. Okay. Thank you, sir. Welcome, participants. The first activity which we are going to undertake is evaluation of the participants for the impact assessment. You will be supplied the performa in Google, and you have to fill it with the fixed time, and it will be submitted. After the training, after 21 days, we will again give the performa and evaluate your knowledge. What is the difference in the knowledge after training, so that we can say that how much this training was having impact upon your PGR knowledge. So I request Ankur and Shwangi to give the Google form to participants and give the time limit.
Sir, Google Form link already shared on the chat box and the WhatsApp.
Okay. So start filling it. How much the time given? 10 minutes?
$15 minutes, I think, enough, sir, for this.
Okay. 15 minutes will be given to you for this filling this performa. देखते रहेंगे प्रेजेंस कितनी है।
71।
हेलो 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 सर।
यस विष्णु जी बोलिए।
सर गूगल फॉर्म परफॉर्मा सेंड व्हाट्सएप चैट पे है सर। गूगल फॉर्म परफॉर्मा। हेलो
It's in chat box. It is in WhatsApp group as well as in chat, both the places.
ओके ओके मैम।
मैं शेयर करूंगा। Then you will use, then you will, I will unsure, then you will show। मैं बता देता हूँ कहने में बदल दी करूँ जैसे यहाँ कर। आप कर।
थोडा येऊ नको थोडा 10 मिनिट थांबा.
So far, we have received only 26 form. So please submit, and you have time. If you need time, then let us know. It is mandatory. It is mandatory, and I'm really surprised to know that only 73% has been joined. 73 has been joined. 70 has been joined, and we have total number of participants, that is 84. So this has been viewed seriously, and this is also required for attendance and for certificate distribution. Anything attendance, I am telling again and again, all attendance will be marked on regular basis. And since it is online training, so we don't have any justification. And one more request to all the participants. If you are using your mobile, try to avoid your unnecessary movement, because it is distracting the speaker as well as all the resource person and everybody. So if you are using through mobile, I will request you have to use a banner in background to avoid the further distraction. This is necessary to fill these Google form what we have submitted, what we have given to you. This is mandatory. Even this is mandatory to all of you. And if you have if you are facing any problem, let us know. And this is the one way we are marking your attendance also. And this is required because at the end of the training, we will also assess you how much impact we have given or how much you have gained.
So till yet only 51 forms we received in Google.
How many, Anku? 51.
51, sir.
So it is a good progress and we hope to receive many
All.
forms, all forms. We can give 15 minutes more, no problem. If somebody has problem, they can communicate through WhatsApp group or can call also.
Dear participant, if you are facing any problem regarding this filling up of Google form, let us know. Please, we have received from Dr. Naresh that he is facing some problem. How to fill this Google form? I have given in the WhatsApp group. The link is recently I posted. You can use that link. Any one of you still facing, please let us know.
This could be,
जैसे ये दे दिया ना अंकुर, आज ये स्क्रीनशॉट ले लूंगा। ओके, इससे क्या हुआ, हमारा भी आ गया सब पार्ट्स।
Ankur, please update how many form you received.
Sir, 54 right now.
54. Dear participant, please fill up the form as early as possible. Because we have further we have lecture from our director sir.
जो पीछे ये लोग जो है
गूगल करके कर रहे हैं चलो, कितना टाइम लगता है फॉर्म भरने में?
Ankur, Ankur, please update how many, Google
56
form you
forms were received till yet.
64,
56.
56. 796. Participant, please raise hand. Those not join, build up the and facing any problem. Let us know.
118 नाही 0
Dear participant, please submit, those who have not submitted this Google form, submit by 12:20. And because we have lecture from our director, sir. Till only 65 participant we have received this Google form. Ankur? Hello, Ankur,
Yes, sir.
update please.
Currently, sir, 68, sir.
68. You just come, close this Google Home.
Close by 12:20, and then we will start our director lecture by 12:30.
Okay, sir.
So-
Definitely.
3 minutes more.
And this is all requested to all the participants, please be online during the director lecture. And he himself is counting attendance. I'm telling you, if he will mark the attendance, and he can force us also. We will not allow then all, whoever is not attending the participants. Whoever is not attending the meeting, we will not allow those participants further. And during the lecture, there are 2-3 suggestions till now I am giving you. First, I will introduce not I will introduce, everybody will introduce in respective sessions the resource person. After that, they will give the lecture. And for any questions, you can put in chat box, either you can ask at the end of the lecture. And all questions should be very quick, clear in the meaning, and it should be, related with our lecture topic. And during the question session, at the end of the lecture, you can raise your hand also using the symbols given in that Zoom. Please take care. Ankur, please close the Google Form process. I don't entertain anybody now.
The form is closed, sir, from my side. Total 71 response we received till here, sure.
Sir, 2 minutes, sir, please. Minutes, sir.
Yes.
2 minutes, sir.
Yes, madam.
Say something. You
Yeah.
Please
Please unmute yourself.
unmute yourself, please. First unmute. Yeah, हम कर सकते हैं क्या? अंकुर किसके पास है होस्ट?
Yes, sir.
अनम्यूट अनम्यूट
Dr. Bangara.
you want to say something?
Participant name, sir.
Dr. Bangarama.
कर्नाटका से यस मुझे फोन भी किया उन्होंने
Madam, please unmute yourself first, then speak.
कमेंट सुनिए ना राइटिंग में
तो किसको unmute?
हेलो। अच्छा अच्छा सर 6 की उम्मीद थी 6 आवाज तो न्यूज में 6 बता रहे सर बढ़िया है सर थैंक यू
शेयर करने के लिए our director has joined. Ankur, please share this. So good afternoon, everyone here. I really feel privileged to have an opportunity to introduce our respected director, Dr. GP Singh sir. Dr. Gyanin Pratap Singh, a well-known wheat breeder, took over the charge of Director of ICR NBPCR New Delhi on 28th of October 2022. Prior to this, he was leading the ICR Indian Institute of Wheat and Barley Research Karnal since 2016. Dr. GP Singh has a vast experience of about 3 decades working in ICR. He has been a wheat breeder at ICR IRI as a senior scientist and principal scientist with a focus on abiotic stress and physiological phenotyping with the help of marker-assisted methodology. His enormous contributions have been recognized with many awards and honors, including Dr. Rafi Ahmad Kidwai Award 2015, Dr. Nanaji Deshmukh Team Award, PGRI Gene Stewardship Award, Dr. A.B. Joshi Memorial Award, Dr. Amrit Singh Cheema Memorial Award, Agricultural Leadership Award by National Education Empowerment and Development Foundation, Outlook Outstanding Scientist Award, Dr. B.P. Pal Gold Medal Award, V.S. Mathur Award, many more. Dr. Singh is a fellow of National Academy of Agricultural Sciences, National Academy of Science, Indian National Science Academy, Society of Advancement of Wheat Research, Indian Society of Genetics and Plant Breeding, Society for Scientific Development in Agriculture and Technology. Evidently, his credential and accomplishment in wheat development garnered him the popular tag of Wheat Man of India. Nothing can be better than to start our training with Dr. G.P. Singh lecture, an accomplished researcher and visionary leader. So I request our director to share his presentation. Sir, please. Ankur, unshare.
Thank you very much. Good afternoon to every participants, colleagues from NBPGR. It's a matter of great pleasure for me to deliver a lecture, the beginning of this training program, and probably it will be much fruitful when we interact also, provided the time permits us so. So I shall begin this. Is it coming on the screen?
Yes, sir.
Yes, sir.
Presentation view mode.
Now it is in presentation view mode.
Yes, sir.
So, as you know, the PGR resources are becoming more and more important nowadays. And people are talking about this because of several reasons. And since NVPGR is the nodal point of all the PGR activities in the country and also the interaction with the international bodies, our role becomes very, very important. And when we see the total utilization of out of 374,000 known plant species, what we are using is only 7,000 which are being utilized for catering the need of human in terms of calorie intake, and therefore there is a vast proportion of the PGR which is still untapped, and there is a good scope of tapping these PGR resources. It's not moving actually. Okay, so India being a gene-rich region, 4 out of the 34 hotspots of the biodiversity, and one of the 12 world's mega biodiversity country, India is, and therefore we are very much rich in this, particularly in the case of India. When we travel Western Ghats, the hottest hotspot, we have more than 1,720 endemic plant species, 162 breeds of domesticated animals. And when we talk of biodiversity, it is not only plant biodiversity, but animal biodiversity, microbe biodiversity, and therefore, traditionally, India has been rich in this, and the question is how much we can use this. We have been an important source of origin of many crops, particularly rice, banana, cucumber, chickpea, sugarcane, mango, bean, eggplant, mustard, citrus, and so many others. And we have been secondary center of origin for several crops which have originated elsewhere, to name them wheat, rye, grape, apple, apricot, and several horticultural crops are there, which originated in different regions but have a secondary source of genetic diversity in India. And when we talk of India's contribution to the global biodiversity, probably we have made much more contribution by contributing around 8.25%. And when we talk of the plants, our contribution to the world's is 11.8%. But we need to still, increase this contribution to show our presence, to show our strength in terms of plant genetic resources. Now, India has been signatory to almost all the commitments to the plant protect biodiversity, and to name some of them India is a signatory to Convention on Biological Diversity, which was signed on 5th June 1992. And then we signed also Cartagena Protocol, And we are also a very active member in International Treaty on Plant Genetic Resources for Food and Agriculture. This treaty entered into force on 29 January 2004. We are also signatory to Nagoya Protocol and on May 11, 2011 and ratified on 9th October 2012. We are also having Geographical Indications of Goods Registration and Protection Act 1999. In India, we have Biological Diversity Act effective from April 2004. And to name one of the most important acts which has been passed by the Parliament is the Protection of Plant Varieties and Farmers' Right Act 2001, which came into force from January 2005. I would like to mention here is that India is the only country in the world which has protected the right of the farmers. And in the recently concluded General Body meeting, 9th General Body meeting, even the International Treaty on Plant Genetic Resources on Food and Agriculture also took cognizance of this plan, the farmers' right. Next year or this year only, we are going to have an international symposium on farmers' right. Now, when we see already India is #1 country, which has been reported recently in terms of population ahead of China, which was expected to go beyond China by 2030, but we are already in 2023 ahead of China. But we would like to stabilize the population and also to utilize these PGRs producing enough food for the world's population. And this can only be done by utilizing the novel genetic resources which have not been so far utilized. And we will depend on the research into everything from high-tech seeds to low-tech farming practices. This slide is very important, I would say. This has predicted by IPCC in 2014. And this projects the situation that will arise in 2080 onwards. And you can see here, global reports indicate a loss of 10 to 40% in crop production by 21st century because of impact on climate change. India is now being projected as one of the major countries which is likely to suffer more in terms of climate variations that may happen in coming time. Now, United Nations sustainability goals are there. I would like to mention here that 17 goals, global goals are there with 169 targets. And when we talk of this, actually, some of the goals are very important in terms of catering the need of the population, which is growing. Like there should be no poverty, zero hunger, and so on and so forth. You can see here the agricultural biodiversity comprised of resilient livelihood, healthy diets and nutrition, and each of the United Nations sustainability goals are mentioned. For example, for healthy diets and nutrition, these SDG 2, 3, and 12 are very, very important. For biodiversity conservation, we have 14 and 15, which involves both animal and plant biodiversity. Climate change is also very important, which is covered in SDG goal 13. And one of the most important goals are sustainable land use, agriculture and forestry, which is covered under 12, 14, and 15. Now, many of you might be aware that 2006, a very important international conference took place in Delhi. And followed by this conference, as a result of this, the Delhi Declaration was made, and which involved complementary conservation strategies, awareness and capacity building, exchange, instrument and biosecurity, frontier technologies for sustainable use. Traditional knowledge is very important for India because India has been projected as one of the countries which has a lot of traditional knowledge. And the question is how much of this traditional knowledge we had been utilizing and tapping. So this is one of the important components of Delhi Declaration on Agrobiodiversity, and participation of private organizations in society, ecosystem services, and agrobiodiversity index. So you can see here, we are following almost all of them except the 9th and the 8th one, which still to be implemented. Now, when we talk of growing, ever growing population, which is likely to be stabilized by 2050, what are our strategy to meet out the population demand in terms of food and calorie intake? And therefore, we have projected in our perspective plan and several other forum novel pre-breeding methodologies. Fundamental reinforcement of the entire plant breeding chain is one of the important and vital part for sustainable system for global food security. And when we talk of pre-breeding trait development, interspecific hybridization involving land races, and as I mentioned earlier, untapped germplasm, also interspecific hybridization and intergeneric hybridization. Now, when we talk of wheat, out of the total genes which have been catalogued through the international system, as you can see here for leaf rust, stripe rust, stem rust, powdery mildew, cereal nematode, you can see many of them came from alien sources. And same way in case of rice also, the genes for different diseases have come from alien sources. But the question is, the contribution from alien sources is not as much as we would expect to have been. And in coming time, in order to meet out the challenges, the contribution from wild relatives become very, very important. The Indian National Genetic Resource Management System in India is actually one of the oldest system in the world, and is most elaborate. And as you can see here, not only we have for plant genetic resources with the headquarters from New Delhi, but we have also at National Bureau of Animal Genetic Resources at Karnal. For fish resources, we have at Lucknow. For microbes, we have at Mau. And for agriculturally important insects, we have at Bangalore. Now, when we talk of PGR, PGR has been one of the backbones of agriculture in most recent times, because it is treasure house for one of the most rich sources of genes. And ICR NBPGR has been in service since 1976 by conserving the gene bank maintenance, then conservation, evaluation, and utilization. So you can see the role of NBPGR in food security is very important, and it is done through the vehicle of germplasm / gene conservation. And we have several aspects of it, like plant quarantine and health testing for pest-free conservation, PGR acquisitions through exploration and import, long-term conservation in terms of National Gene Bank, and many of you might have visited. At the moment, we have 484,000 accessions which are stored in the gene bank. We are also planning to have a safety duplicate in coming time, so that in case of any exigency, we have at least one copy conserved elsewhere other than Delhi. And we have policy, the PGR policy and PGR informatics unit, and characterization and evaluation for different traits. These are some of the stalwarts which have been working in NBPGR. Actually, I would like to pay our homage to Dr. Harbhajan Singh, who was one of the most important persons who started PGR activities, followed by Dr. K.L. Mehra. And the modern time, NBPGR and its stations came into enforcement when Dr. R.S. Paroda took over as the director of the National Bureau of Plant Genetic Resources. We have more than 10 regional stations. And we have also national active germplasm sites at various places pan-India. You can see here we have in Srinagar, Simla, Jodhpur, Hyderabad, Thrissur, Akola, Cuttack, Ranchi, Shillong, and Bhowali, catering to different agroclimatic conditions with headquarters at New Delhi. And you can see from establishment in 1976 to 1985, we have reached up to stage of 2023, where we have a modernized gene bank, we have high throughput genomic unit, trace-specific evaluation, utilization of land races, crop wild relatives, and genomic resources, and we are ever growing since its establishment. As I mentioned, you can see the number of accessions actually which are there. A total of 283 exploration took place, and we have conserved these accessions which were found out in the exploration process. Our exploration actually mainly concentrated in the northeastern part of India, which was highly difficult and disturbed area apart from Kashmir, and in the Naxalite inflicted areas of Chhattisgarh. So you can see the efforts of our scientific staff and technical support staff doing enormous job in going for exploration in these areas. We have also a unique and potential germplasm first time collected through these explorations. You can see here traditional tuberous vegetable of Khasi and Janithia tribes in Meghalaya, and from Arunachal Pradesh and several other parts of the country. And you can see from this picture, our staff is working in a very difficult situation when they go for exploration. We also collected novel germplasm, carotenoid-rich cucumber from Manipur and Mizoram, brown-knitted cucumber from Meghalaya. You can see tree cotton from Mizoram, light brown linted tree cotton from Tripura, cut leaf mustard from Arunachal Pradesh. Recently, our exploration unit went to Assam, and they brought several kinds of citrus species, which I have not seen actually. I was very surprised to see this because we have such a rich diversity of citrus in northeastern part of the country. We have also potential new economic plants identified and demo plots established in different parts of the country. For example, cut flower and several other species. You can see in mango also several oryza new species have been identified, catalogued. And we are also geo-referencing and mapping of gap from the collected germplasm, and we are maintaining, in fact, a national herbarium of cultivated plants, which is one of the most important activities of National Bureau of Plant Genetic Resources for allium species, black gram, and several minor millets. We have also a web page for crop wild relatives. Now, introduction and exchange has been one of the most important activities of National Bureau of Plant Genetic Resources because all the material which is being imported is done through NVPGR, which is a nodal point, and exchange of PGR facilitated for more than 105 countries and international agriculture research centers. Every year, over 20,000 accessions of germplasm and 50,000 samples of trial material received from utilization. These trial materials are coming from CG centers and it is being distributed to different evaluating centers. We have also more than 420,000 accessions supplied to different intenders for use in national program. We have a small unit of germplasm exchange and policy unit where import is being facilitated from CG and other countries. And this activity, we are not only giving to the NARS partners, but also for the private companies dealing with crop improvement activities. And we are also generating resources out of this. You can see here promising resources and accessions from new crop species brought through introduction in wheat, in French bean, soybean, sunflower, cucumber, carotenoid, pomegranate, improved varieties in buckwheat. New species have been also imported from US. The list is very, very long. I cannot read out. I think probably Kuldeep will be providing this presentation to each and every participant as a matter of record so that they can refer this in coming time. Now, when we import the material from outside outside India, quarantine becomes most important activities of our institute, because we cannot permit any weed or nematode or any pathogen coming into India which is not present here at the moment. And therefore, this is an enormous activities, and we involve several new techniques, treatments which have been developed and standardized for detection of pest and salvaging of infected, contaminated PGR, which is coming through as a routine affair from through introduction. Quarantine processing of germplasm under exchange during 2021-22, you can see here now 1,37,000 accessions were imported at Delhi and 16,000 accessions at Hyderabad, which is a very important station of NBPGR. And this is an enormous activity. And you can see, lot of material was detected with having some or the other pathogen which was not present in India. And we take a standard operating procedure to damage and reject, salvage these kinds of accessions. And a post-entry quarantine check is also made. The total of 54 PEQ inspections were conducted through NBPGR, and a total of 82,000 samples of various crops were inspected. When we talk of probable annual losses because of these pests or weed or any other foreign material which is not present in India, you can see it could be enormous proportion, and probably the annual loss could be 479 million Indian rupee, and yield losses can also be to a great extent when we assume that a total loss is 0.1%. But when we talk in real terms, the situation can be much higher in most of the cases. Now, the second major impact activity of National Bureau of Plant Genetic Resources is Exchange and Policy Unit. It plays a very vital role on the international discussions, particularly in recently concluded preparatory meeting for GB9 of ITP GFRA, which was conducted in September 24, 2022. It will be conducted. Comments, inputs on Biological Diversity Amendment Bill 2021, which is pending with the Parliament, and several of the activities. Our unit is playing a very vital role in suggesting to the government bodies for the policy and other impacts. This is one of the important activities which was held in the month of September. India hosted GB9 meeting of ITP GFRA, and this was a very successful event in many terms. For the first time, ITP GFRA acknowledged the farmers' right in this meeting, and new international symposium is proposed in November 2023 on farmers' right. Also, Indian delegation led the deliberation on various agenda items, and GB9 re-establishes an ad hoc open-ended working group to enhance the functions of MLS. India is also, as a matter of pride, I would take pride in this, telling you that India is elected as a co-chair and we lead the process along with Australia. Now, conservation is one of the important activities, and conservation involves two-pronged activities where we do for dynamic conservation and also static conservation. In dynamic conservation, ex situ, in situ, on-farm community gene bank, field gene banks are very important activities, and many of you might have visited our gene bank here in headquarters in NBPGR or field gene banks at various regional stations, and you might be aware of this. But the new impact of this is a static ex situ gene bank where permafrost is being contemplated as one of the activities which will come up in coming time, and India could have one of the safety duplicates. Presently, only one safety duplicate is existing in Norway, in Svalbard, but India is likely to have another safety duplicate for our own use. And this could be done through permafrost and gene banks, seeds, also in vitro tissue culture, cryopreservation, long-term storage, medium-term storage. It's all activities which are coming. It's going on also, and we would contemplate to improve all these facilities in coming time. As I mentioned you, a total of 462,982 germplasm exist for different crop plants, which involves in the national gene. And when we talk of crop wild relatives, you can see here for wheat, rice, sesame, solanum, amaranthus, and several of the crops we have accessions which are from wild relatives. A total of 9,174 accessions belonging to 662 genera are there in the gene bank, which could be one of the wealth in the in coming time to utilize in the crop improvement program. In vitro gene bank is also there, where we store with the help of cryo tanks and tissue culture techniques. Tissue culture technique is particularly utilized in terms of tropical fruits like banana, temperate and minor fruits, tuber crops, bulb and other crops, medicinal and aromatic plants, spices and industrial crop. And we have in total roughly around 36,000 cultures which are there in this in vitro gene bank. Okay, there is some technical glitches. Somehow these slides are not moving actually. So you can see here we also do cryobank in field gene banks and also repatriation of in vitro conserved taro germplants to farmers' field. And this could be one of the most important activities in coming time. We also utilize our Isapur farm for doing all.
Sir, you may like to use the arrow button, sir.
Arrow, I am using Sunil, but it's not moving actually.
Somebody's coming there. Anyways.
मूव नहीं कर रहा है. इसको स्टॉप कर दें. ठीक है, मेरे ख्याल से मूव करेगा. In situ conservation of wild rice Rufi Pogon species in Sonitpur, Assam is one of the important activity. In 2018, ICR NBPGR located good population of Rufi Pogon in a pond located near Pagla Bil Gaon between Borjuli Chai and Bagan and in Sonipur district of Assam. Recently, we have also entered into agreement with National National Rainfed Authority for undertaking this activity. Wild rice populations like pure crop available in this pond is so far neither seen anywhere nor reported. So this is one of the important treasures we have, and we'll be taking up this activity in times to come. Now, legal protection is also existing for in situ conservation of the sites in Assam. On 7th July 2021, Bojuli Tea State, owner of the land, gave NOC for conservation of the site. This legal protection in the Biological Diversity Act 2002 was essential to actually maintain the site and conserve the species in nature for a longer period of adaptive and evolutionary process. Stakeholders like NBPGR, Assam State Biodiversity Board, and several other National Diversity Board are involved in this, and they made concentrated effort to declare this as biological BHS. On 10th of August 2020, the government of Assam has declared, notified through Gazette, this site a biodiversity heritage site for wildlife. And this is one of the major activities and important activities. As I mentioned you, we have recently entered into agreement with NARA, the National Rainfed Area Authority, has agreed to fund this project, and hopefully this activity will further elaborate. Other initiatives on in situ conservation also been happening for crop wild relatives. For example, habitat of guava in Thal, Pithoragarh, Uttarakhand district, and several of the areas in Dantewara, Chhattisgarh. We are undertaking this, and you can see the pictures are very, very clear how difficult it is for our staff to go for these kinds of activities in this area. These are also not only geographically very difficult areas, but also law and order situations also do exist in these places. And therefore, my all compliments to my colleagues who are going for this. You can see from this picture utilization of gene bank genetic resources. And we had been doing these activities in the past also, and you can see the percentage of accessions which have been evaluated over a period of time, and you can see the number of accessions which have not been evaluated. So this is actually ongoing activity. We need to hasten up this process. More and more genotypes need to be characterized on the basis of their evaluation, so that unlocking the genetic potential stored in this gene bank is properly utilized, and it is our responsibility towards future generation. New challenges which would arise, and hence new opportunities also for NBPGR, because once there is challenge, you think in a different way. And therefore, when we talk of climate change, when we talk of natural farming and other activities, we need to have different kinds of changes. For example, genomic could be one of the important activities in characterizing and evaluating this, not only the accessions, but also finding duplicates which are existing in our gene bank, so that they can be removed through characterization and evaluation. And Dr. Sunil Archak is very active in this field. He is developing PGR informatics. This has led to a paradigm shift in the approach focused on enhanced utilization. In times to come, we will be more depending on genomics and in bioinformatics to make PGR more accessible, more useful to the people who involve this in the crop improvement program. As you can see here, flow of genes from gene bank to breeder is very important activity. It has to go from gene bank to breeder. Breeder in turn will utilize this and ultimately develop a variety which is climate resilient, which is high productive, and it is economically more useful to the farmers. And then only our activity ends. And this is an ongoing activity, will go on and on from time to come. In recent past, we have taken a massive exercise of evaluating more than 20,000 wheat accessions. And this actually has led to development of a core set and a mini core set, a reference set for terminal heat tolerance and wheat, which is something around 600 accessions, starting from 22,000 to 600 is, and representing the entire genetic diversity is a great achievement. I would request all the NARS partners to be in touch with our people who have done this, and we are ready to supply this material for further utilization. We've also taken up chickpea evaluation and characterization activities. More than 18,000 accessions were evaluated, characterized for different descriptors, and large-scale phenotypic and genotypic characterization and evaluation of an NGP material started for various crops. Presently, more than 25,000 sorghum accessions are now being evaluated at Akola Center, and probably we are going to hold a field day in Akola. The tentative dates are March 10th and 11th, and all of you are invited to join there to see the amount of diversity. We are also taking up characterization course and development of wheat, apart from wheat, other crops like rice, cowpea, linseed. Development of core set and mini core set is very, very important in terms of representation of entire genetic diversity, because you cannot go on and finding, although our gene bank is automated, you can retrieve the information at a very faster speed now. But once we develop core set and followed by mini core set, you have treasure in a small box which can be utilized. Multiplication and characterization of crop germplasm for genotyping and conduct of multi-environmental trial trade is being now going on. We are very thankful to the Department of Biotechnology providing very generous fund for this. As you can see, for wheat and other sesame, other crops, evaluation process is taking place. In next month onwards, probably the monitoring of this will go on. We have also done phenotyping of 2,634 accessions of linseed germplasm for agromorphological traits and characterization and multiplication of 700 Niger germplasm. Various wild species have also been characterized and evaluated. The entire information is there in our gene bank retrieval system. Anybody who is involved in these species improvement program probably can contact us, and we will be very readily providing the information to them for improving the crop improvement activities. Now, many a time, most of you probably belong to the crop improvement discipline, and you know that it is sometimes become very, very difficult for crossing between wild relative and the agronomic type, and therefore pre-breeding activities become very important for genetic enhancement. And we had been doing this activities. 65,000 accessions have been already supplied from NBPGR to another national active germplasm site in 4 years, preceding 4 years, excluding the germplasm which has been shared in collaborative projects. So you can see we have developed pre-breeding material from pepper, okra, brinjal, lentil, vigna, and the trait of interest has also been clearly stated here. You can use this for improvement of these crops. The entire germplasm, the new activity of biochemical characterization has been going on in National Bureau of Plant Genetic Resources since last maybe 5-6 years. And you can see in various accessions, we had also been predicting, for example, in case of rice, protein amylase, in maize, oil content and protein content, linseed also, black gram, barnyard millet, vegetable amaranth. Different quality parameters have been clearly quantified, which can be used not only for yield improvement, but also for quality improvement. Because from particularly in case of some crops like wheat and rice, where we have gone from shortage to surplus, now quality becomes very, very important. And recently, India exported more than 14 million tons of wheat. This happened only because in the last 2 decades or 1 decade, the quality of Indian wheat enormously improved. When we compare with that of European wheat or Australian wheat or South American wheat. These are some of the unique trait-specific germplasms which have been registered. Recently, we had 49th meeting of germplasm registration committee. We are contemplating to have some modifications in the guidelines for germplasm, which will be shortly, published. But as of now, more than 1,900 germplasm has been registered. This is openly available to all of them, all of you, which if you want to use in different crop species, cereal, pseudo-cereal, millet, fiber, forage crops, grain, legume, vegetable, you name any crop, we have genetic lines which are registered, protected for IPR, and that can be used for, we had a publication also continuously, which clearly depicts the trait of economic importance for which these genetic lines have been registered. You can see here, yard-long bean varieties have been developed. Varieties released from germplasm collection in coriander. Varieties have been released by different universities in cluster bean also. As per the Government of India notification, the DNA profiling for the release of the varieties through Gazette notification has been made mandatory for quite some time now. And NBPGR is providing a great effort in this regard. Whosoever requests us for DNA profiling, we are doing it. Although many of the crop commodity-based institutions are also doing it. But I am emphasizing on this only because, many of the organizations, if they don't have facility, to DNA profile their variety, we are ready to use on a very nominal charges. And if you are from NARS, probably that is also exempted. So all of you are welcome. You give your material, we will DNA profile this, and this is mandatory for crop variety release, and therefore you can provide the material, we'll do the job for you. We have National Genomic Resource Repository, a total of 9,000 genomic resources representing 6 species are being conserved at -80°C and also on -196°C under the National Genomic Resource Repository, representing several crop species, be it brassica, chili, chickpea, cotton, linseed, rice, probably one of the largest, sorghum, and sesame, vigna, sunflower, through cryotanks and deep freezers. Development and utilization of genomic resources is very important because it becomes immaterial when you develop genomic data and they are not utilized. So we have been doing in several of the crop plants, SNP discovery in lentil through whole genome resequencing in ridge gourd, and this can be utilized for further developmental and improvement activities. And phylogenetic trait for 96 lentil accessions based on 1,27,000 SNPs had also been developed. This can be used. Likewise, in rice also, SNP and SSR markers have been utilized to develop this genomic information. You can see here generation of genomic resources and identification of gene and pathways associated with grain size in Vigna has been conducted. I would like to tell you that based on the genomic information, of course, done at IIWBR and in collaboration with NBPGR, we have been able to identify one new gene, LR80, which has been catalogued. This is only the 5th gene which has been catalogued from India, and this is a great activity. I would like to promote this activity at NBPGR so that all the material, depending on their genomic information available already in major crops, we can go for trait discovery, gene discovery, and other activities. Here you can see an association panel of 135 diverse wheat genotypes was genotypes using 35K exam SNP array, and it's a huge information which is available that can be utilized in crop improvement activity. As a matter of fact, 10 major QTLs with significant effect were identified for salt tolerance, namely leaf chlorophyll content, sodium and potassium concentration and ratio. And this could be one of the important starting point for major change in improvement activities for salt tolerance, because we have 1 to 3 million hectares of land, in case of wheat particularly, which is being grown in salt affected soils, and this can lead to major varietal development program, and it can lead to major productivity enhancement in these areas where productivity is very, very low. You can see in rice also, and also this has been done. Linseed is another crop where 23 significant QTNs have been identified for flowering time, physiological maturity, seed weight, and plant height. Development and validation of cast marker associated with spot blotch in QTL identified through GWAS in bread wheat, because spot blotch is becoming more and more important, particularly in the eastern part of our country, Indo-Gangetic plains. And this is a complex kind of trait which is governed by several QTLs and genes, minor genes. And this GWAS was performed on an association panel of 294 wheat accessions, probably received from CIMIT, which are their core set, and genotypic evaluation and testing panel using cast marker was done. And this led to generation of information which can be definitely utilized for improvement activities, and also in linseed, new gene from early and late flowering maturity linseed accessions was identified. Now, GM detection and diagnostic is an important activity of exchange. We have a ISO certified facility in NBPGR, and ICR NBPGR is the nodal agency to issue import permits under and undertake quarantine processing of transgenic as per the Government of India notification. And more than 245 consignment of 15 crops were imported through ICR and NBPGR. Whosoever requests for GM detection facilities for molecular testing and evaluation of these transgenic material exist in our campus, and we can do it readily on the basis of payments of I would say very nominal fee probably for GM detection. This is one of the major database which has been developed by our colleague Dr. Sunil Archak for linseed, for grain amaranth. I'm sorry. This is one of the genetic resources on grain amaranth comprising of SSR, SNP, and microRNAs. And I guess probably this could be taught in the practical classes how to access these genomic resources database, not only for amaranth but other crops also. As I mentioned earlier, in coming time, genomic resources and bioinformatic tools are going to be one of the major vehicles for crop improvement program. And most of us, probably 50 plus people, don't actually are familiar with these activities. Younger scientific community of our country is more and more aware of this, but we are actually developing program of development of bioinformatic tools. A germplasm duplicate identification and removal tool was developed by the group of Sunil Archak, GDAT. This can easily detect duplicates and remove from the gene bank. This could be one of the major activities in conserving and saving resources we have. GDAT has been validated for duplicate identification in many crops on identity by state analysis using SNP data. And this has been extensively tested in 1,133 genotypes of different triticum species. So this is very well validated and functioning very well. And probably you will get a good course of learning from this exercise. PGR informatics brings transformation. That's a very bold and statement. And as of now, during last 10 days, PGR is assuming greater and greater importance, and an organized digital information system provides fair and just opportunity for all to access. So the major philosophy behind this is the access of the material and benefit sharing, as I told you earlier. So through these, you can see the left picture and the right picture. What was the condition maybe 25 years back, and what is the condition right now, where we can have informatics system for retrieval and utilization system. So this is one of the changes which is happening globally. We are also in line with that. Now, the last question which is being asked is why PGR informatics has come into limelight. This has happened only because during last decade or so, or maybe since 2004 onwards, when several of the international treaty in India and elsewhere came into force, national and international act and treaties for PGRFA, various international agreements are coming also in force. Availability of information in text, image, and map videos, technologies to record, link, and archive such diverse type of information, ever increasing power of computers, internet to facilitate access and retrieve. So this could be one of the driving force for PGR utilization for crop improvement activities, and assuming a greater importance there. We have developed several of the databases and applications. It is mentioned here: passport database, import database, gene bank database, cryobank, characterization database, and lately, in vitro gene bank database. As far as applications are concerned, PGR portal, gene bank dashboard, cryo dashboard, PGR map, PGR claim, GRIS, e-herbarium, CWR. These are all developed at National Bureau of Plant Genetic Resources, thanks to our team led by Dr. Sunil Archak and his group. from conservation. And we need to improve this in times to come, and we need to include all and every extensions and crops which are being stored in our gene bank. 6 databases, 10 applications, 4 mobile apps. That's the summary of what NBPGR has in terms of bioinformatics system. NBPGR website has 1 lakh hits per year, and it will increase, I guess, in times to come. Some of the important activities is PGR Conservation and Awareness Program. PGR Conservation and Awareness Program conducted and farmers are sensitized, particularly in those areas where we have rich heritage of biological diversity. And this is being done through the Council's funded program of Tribal Subplan and SC Subplan. And tomorrow also in West Bengal, one of our team is visiting to make aware the farmers about their rich heritage. And these are the activities which will go on, because we need to educate our farmers on this. And this could be one of our most important activities in times to come. When we talk of farmers' right, then we need to educate the farmers also about their rights and their heritage they have. So at the end, I would like to thank all the scientific community who is working in NBPGR and elsewhere in state agricultural universities, ICR institutions, in commodity-based institution. Without help of these people, it will not be possible to progress on in terms of plant genetic resources, its conservation, utilization, and ultimately, the benefit of the farmers coming through this vehicle. Thank you very much.
Thank you, sir, for your. Please give a big hand to our respected director. Thank you, sir, for presenting and informating a detailed overview of ICR and PGR activities. I am sure that all the participants must be benefited with your words. So, sir, if you permit, we will take some questions, if any. Sir. Sir, if you permit, we will take some questions, 1 or 2 questions, if any.
Or else you can have in chat box.
We have not received, sir, any question in chat box.
Okay.
If I request participants, if you wish to ask anything related to PGR management, just 1 or 2 questions, either raise your hand or put in chat box.
I don't think there is anybody who would like to. Anyway, this is the beginning actually. Once they go through the course of this training program, they have more and more queries. Yes. And probably through course of the time, their knowledge will be updated at greater length. So I think if they don't have a question right now, I think we can take up it later also.
Sir, one question some Vinod Kumar has asked. Sir, how can we get germplasm from NVPGR? So maybe in next session we'll answer this query related to exchange.
आप बता दीजिए, there is an MTA process of MTA, material transfer agreement. You give the accession number or anything you have, sign an MTA, and we'll provide that. It's a very simplified process.
Mm-hmm.
If you want to have from other country, I think you have to go through different channel. If you want to have material from our own gene bank, it's a simplified process.
Another question, sir, can we also join from SAU's for explorations?
In fact, I would promote that. In the areas where we are not very well aware with inhospitable conditions, if the local people through state agricultural universities also joined, I think that would be a great idea. Probably subject to the financial availability at that moment of time. But this will be a very nice idea if we have local people also. Although we are utilizing KVKs and other people, but I think we don't have any objection to that. But the collections would belong to NBPGR as a national treasure.
No, so many questions are there. So shall you take up, sir? Other questions also.
आप देख लीजिए if your time permits.
Yeah.
No, if they deal with specific lectures which are already planned,
yes,
so those can be taken there.
Yeah,
Right, so
that can be done.
we have subject specific lectures, so we'll take in due course of time.
Yes.
Mhm.
Now, I think we have to thank our honorable director for sparing the valuable time and start a great start of the training with his lecture. So thank you very much, sir, for your time. And,
Thank you. Wish you a very good training course for 21 days.
Thank
Thank
you,
you
sir.
very much, sir. Thanks.
Thank you, sir.
Thank you.
So now we will break for lunch and we will join again at 2:30 p.m. as per the schedule we have. Thank you. Okay.
With protection of plant variety and farmer rights authority. She's also involved in the She's also involved in preparing technical inputs and developing countries' positions for negotiations at different international meetings related to genetic resources. Recently, as Dr. G.P. Singh, our director, has told in their inaugural lecture, she represented, Dr. Pratibha Brahmi is one of the member from Indian representation. She represented India at general body meetings of ITPCRFA. She has attended advanced training on genetic resources and IPRs at University of Agriculture Science, Sweden. And she has also published more than 100, including peer-reviewed, more than 100 publications, including peer-reviewed articles, book chapters, manuals, edited books, and policy papers. For any maintenance of law and order of the country, we need some set of the principles that we can call constitution. Similarly, the policy is also required for PGR management. We have to set some principles, some policy framework. So Dr. Pratibha Brahmi is looking after that activity in NBPGR. In fact, we can say from ICR side. So, madam, floor is yours. I am sharing these slides. You can.
Thank you, Kuldeep, for this very generous introduction. I'll just share my screen. Yes.
It's a visible, ma'am. Please keep on slideshow.
Is that okay?
Okay, that's fine, ma'am. Fine, absolutely.
You can hear me also clearly?
Yeah, sure. It's clear.
Okay, so this is the, I think, very beginning of your training program, and I'll be talking about policy issues in PGR management. As you all know that policy, like Kuldeep just now said, policy is in English, translation, if you see, it means policy means to way of doing things. So regarding the management of genetic resources, there are many policies at international and national level which govern the access, exchange. I have listed what are the international frameworks which affect management of genetic resources. That means first one is the UN, the United Nations FAO, Food and Agriculture Organization, because we are working in the agriculture sector. So these are the policies which we have to look or forward in the UN FAO. Whatever policies they are making regarding agriculture, food security, that we have to keep in mind. And the FAO also has a commission on genetic resources for food and agriculture. Basically, we have to understand the difference between genetic resources which are for food and agriculture and which are general genetic resources or bioresources, you can say. I will come in the next few slides. What is the difference between these type of genetic resources? Then we have also the international treaties which are signed by different countries, and when a particular quorum of countries sign that treaty, that becomes obligation on that country to make rules and regulations with reference to these international treaties. So these include the CBD, the International Treaty on PGRFA, and also some guidelines regarding IPRs are given in the WTO's TRIPS agreement. I will explain everything in detail in the next few slides. Here I am just listing these, what are these policies which affect. Then in the background, we have supporting policies which relate to environmental laws, water bodies management, sea management, trade and IPRs. Then we have also the general guidelines which are made at the international level. These are called as the global plans of action. And these are guidelines on how you manage your genetic resources. Okay. So, and also based on these international treaties or rules or regulations, we have the national management. These responsibilities are based on international frameworks. And also the basic principle is that all countries have sovereign rights over the resources. That means they make rules and regulations on how they manage the genetic resources. And based on these principles, we have the national laws and regulations. And the third thing we have to keep in mind are the mandated activities of the institute and the institutional policy. Because each university or an institute or like council, they have their institutional policies also for dealing with these laws and regulations and how they manage them. So like I was saying, the genetic resources, what we deal under agriculture, and especially talking of the plants. So these are called as the plant genetic resources for food and agriculture. That means genetic resources which are important for food and agriculture. So this is different from the general biodiversity, and these are the basic 5 differences. One is that these resources have been developed over centuries by the cultivators, by the farmers, by the local communities. They have grown them over generations, and because of that, they have developed certain distinctive properties or qualities which were suitable for our consumption or nutritional security. So these cultivators have developed these over centuries. Many millennia have gone into developing our local land races, which have now been used into modern plant breeding. And they differ from the naturally occurring diversity. Obviously, when we select our what is suitable for us, what is good for us, so some of the characters they lose which were there in the wild, but to our benefit. So they are different from naturally occurring diversity, and they are mainly managed by humans. That means without human intervention, we cannot cultivate these varieties which have been developed over centuries. And also these are evolving continuously, because we are breeding them, we are using them, we are putting them into different agro ecosystems. So each will develop its own unique trait. And also when we talk of the genetic resources, there is an interdependence. That means countries, they are interdependent on each other for getting or managing these resources, simply because the origin of croplands is different from where they are now cultivated. There are many examples of crops, especially which were developed, which evolved or originated in, say, South America. They are now cultivated world over. Similarly, wheat, which is from the mid-Asian countries, it is cultivated all over the world. So for genetic resources which are occurring in wild, from where there is a pool of useful traits, those traits still are available in the origin, the country of origin. So these are available, and interdependence of countries is very much important. That also makes it very important to know what are the policies for management of these genetic resources. So when we come to the access, that means availability and utilization of these resources. So these international policies and laws and regulations, they govern access to germplasm for research as well as commercial utilization. So at the international level, there are three basic, GRFA management. That is, one is the CBD, on which our Biological Diversity Act is based. And then we have the FAO's Commission on Genetic Resources, which has led to the formulation of National Action Plan for Agrobiodiversity. And the WTO TRIPS has led to amendment in our Patent Acts, Protection of Plant Varieties Act, and the GI Act. So access to genetic resources under different regimes, if you say, before 1983, then in 1993 with the adoption of CBD, and then 2004 onwards, that you can say there are three different stages. Before 1983, there was free access to germplasm, and the germplasm, the genetic resources were considered as the common heritage of mankind before 1983. And then came the international undertaking on PGRFA, which was basically to say that since all countries are interdependent on each other, for the world's food security, the exchange of PGRFA should be free. And after that, when CBD came in 1993, there was a regulated access, which was based on the principles of CBD, that is PIC and MAT, which we call it prior informed consent and mutually agreed terms. And the CBD also defined that sovereign rights, the nations have sovereign rights over their genetic resources. So this led to somewhat, regulation of access. Before that, it was not regulated. After 1993, it got regulated. Then when this IUPGR was finally adopted as ITPGRFA in 2004, then it became a multilateral access multilateral access means many countries can access from a small pool of or a big pool of germplasm. I will explain in next slides. So the first one we say is the CBD, which it covers all types of biodiversity. That means everything which is living on this earth, on the biosphere of the earth, is the conservation of all those biodiversity is the objective of CBD. Then how to use these, so the second objective is sustainable use of components. Sustainable means that we make it available to the next generation also. So we should not only sharing of benefits arising from the utilization of genetic resources in a fair and equitable way. That is the third objective of the CBD. And the access of these genetic resources by the national government would be subject to prior informed consent and mutually agreed terms. That means prior means the country who is accessing has to define what use they will put it, and the benefit sharing would be on mutually agreed terms. Then coming to ITPGRFA, this covers all PGRFA, that means plant genetic resources, but the use of genetic resources PGRFA should be only for food and agriculture purposes, and therefore this does not permit access to any PGRFA for non-food or non-agriculture use. That means if a crop has multiple uses, including food use and medicinal use. And the treaty has established a multilateral system of access of selected crops. These the treaty also has set rules for access and benefit sharing of these material, which are occurring in a country both in ex-situ and in situ form, but while respecting the IPRs on these resources. That means the country who's accessing should respect the IPR of that. Okay. So in a tabular form, I have just listed here that under the CBD, there were two more protocols which were discussed. These include the International Protocol on Biosafety, which is popularly known as Cartagena Protocol. And this provides an international framework for trade and protection of environment from adverse effect of products of modern biotechnology, that is GM or any other living modified organisms. If they are exchanged, it should not affect the environment. So this protocol has led to formulation of biosafety guidelines, safety of biotechnology by DBT in 2002 under the Environment Protection Act. So the obligations of this protocol are listed in our guidelines of Environment Act. And this was adopted in January 2000. Then the second protocol under the CBD was the International Regime on ABS, which is also known as the Nagoya Protocol. And to this, for this also, our government has issued a notification in November 2014. This basically deals with the internationally recognized framework for access and equitable sharing of benefits. And it is binding for all parties. And this came into effect in January 2017. This basically talks of how countries should share and the benefits from utilization of genetic resources in an internationally accepted manner. Then this I have already talked that this was adopted by FAO in 2001. And it is legally binding for all the countries that ratify it. And countries that ratify, they will have to bring national laws into their country for conformity to the treaty. And here also I would like to emphasize all the CG centers which are working on, which are international institutions like IRRI, Philippines, working on different crops. They have also signed agreement with the treaty that all their material would be put in formally into the multilateral system of the treaty. So anything you access from these centers is governed by the rules of the treaty. Then based on this WTO TRIPS agreement, we have our Protection of Plant Varieties Act, the Patents Amendment Act, and the Geographical Indications of Goods Act 1999. Here that each member country, they should comply with the minimum standards of protection of IP. And this includes protection of microorganisms, non-biological processes, plant varieties, which meet the protection criteria. And this also came into force in 1995, and it is also legally binding for all WTO members. And if we don't follow this, we can have trade sanctions if we cannot comply with these. Then the other, agreement which affects agricultural exchange is the agreement on application of sanitary and phytosanitary measures, that is called as SPS agreement. And this includes that when we are exchanging food commodities, they should not be harmful to any human or animal life, and the foodborne risks which are carried by these plants, they should not be transferred from one country to another. They should take measures to minimize these. Then also we have another agreement which is based on the exchange of genetic resources, is the International Plant Protection Convention. That means when we are exchanging material, it should be free from diseases. It should not spread to other countries. So this is an instrument which harmonizes the phytosanitary measures used across international borders. And our plant quarantine regulation for import into India is based on the amendments of different when these IPC conventions are adopted. Then we also have the international standard, which is the Union for Protection of Plant Varieties. Although India is not a member, this is to maximize the plant breeding efforts, and also it provides kind of a model for securing protection of plant breeders' right. Although our act is based on the guidelines of UPOV, but India is not a UPOV member. And it is also legally binding to parties which are signatory to UPOV. Then this I was talking of. There is a global under the CGRFA, global plan of actions are defined, and these are gradually covering all genetic resources for food and agriculture. That means plant, animal genetic resources, aquatic genetic resources, and microbial and other invertebrates. And this serves as a framework there is also a code of conduct for plant germplasm collecting. That means if you go and collect in other countries, the germplasm, that how it should be shared with the host country, how you should transport them, what are the responsibilities of collectors. All these are given under these FAO code of conduct. I think other speakers would be talking on these aspects also as well. This I have already talked. Then we come to the national legislations which has implication on access to genetic resources. So we have the Protection of Plant Varieties and Farmers' Right Act. This came into force in 2001. Here there is a system of registration of plant varieties which grants the breeder's right to the developer of that varieties. So here new varieties which can be protected, that means they are given registration under this act, and the registration is based on these 4 principles. That is novel, that means the variety should be new in the market. It should be distinct from other varieties existing in the market. Then it should be stable in its performance, and the plant variety should be uniform in expressing all these traits. And these registration is granted after the PPVFR authority test all these parameters which are claimed in the applications, and for which they conduct certain test, which is called as the DUS test. So after the variety is found new as per the DUS guideline traits, all the parameters are statistically analyzed, only then a variety is registered under this act. And the other thing to notice in this registration process is that when you are developing varieties, you are using multiple lines. You should have all the passport data of all the genetic resources which you have used in the development of new material. And also you should have a proof of lawful acquisition of the material. That means when you acquire these resources, you should have a proof that you have lawfully acquired, you have not stolen, you have not used somebody else's material without the consent or permission of that. So all these documents are required to be. Why I am talking about this is when you are using genetic resources, and we should keep a record how we have obtained them, and also what are the passport data of those genetic resources. So if these are not provided with the applications can be rejected by the PPVFRA. Then I have already talked about the CBD, for which we have the Biological Diversity Act. So that in this act, what is to be clearly noted and propagated also to everybody, that access to biological resources by non-Indians is subject to the approval of the National Biodiversity Authority. That means if any foreign researcher collaborator requests you to share your material, you cannot just simply give it on their request. It has to be approved through a process, which is the National Biodiversity Authority. However, if you have collaborative research projects under which is approved by the government, excuse me. Then under the collaborative projects, you can exchange genetic resources, but your project should be approved by the government, any government, central government or state government. And also, if there are students who are taking their material abroad for research, then they can get approval from NBA for carrying this material with subject to the undertaking by the foreign guide or the researcher that it will not be used for any other purpose. For that, there is a Form B under the at the NBA website, which can be used. Then coming to another act, which is the outcome of WTO, is that some of the goods which are agriculture goods, which have a certain property or a quality which is due to its cultivation in a particular area. These attributes can be linked to that geographical region. So these could be land races or crop specific certain agroclimatic regions of the world, of the country. I have given some examples like the Darjeeling tea, the Malabar cardamom, then Malihabadi mango, or the silk products of like Chanderi, Tassar and Munga. These are all geographical indications which are attributed to certain quality products. And this gives advantage to the owners of these GIs, that they get a premium price in the market, as well as they can continue to re-register there. And this type of right is always granted not to individuals but to a group of cultivators or who register for this GI. Then the plant quarantine regulations for import into India, especially and this also includes import of GMOs. In this, you have to get quarantine clearance of any material which is imported into India, and certain agencies are involved in giving these quarantine clearance. Regulating import of plants into India is governed by this. And the three basic principles of this act, this regulation, is that when you are importing any material into the country, it should accompany with a phytosanitary certificate by the exporting country. And also it is examined in country when it is arrived. And there are certain parts of this act which deal with the list of material which should be coming with additional declaration on the phytosanitary certificate, or certain material which cannot be imported from certain countries. So there are schedules under this quarantine. I think the further speakers will be talking on specifically on this act. This is just for information here. This I have also already talked that there is a national framework for development of genetically modified organisms or resources and on their commercial exploitation. And these guidelines give how these GMOs or LMOs are to be imported and shipped. This is the last one I will say here, is that this seed bill, it is still under revision under with the parliament. This basically talks of import of plant seeds, which should conform to certain minimum standards of germination, genetic and physical purity. And they will basically be dealing with all the seeds which is available to commercially to the users, that is farmers, that they should have minimum standards of purity and germination. So this is the summary slide. I would say that how different activities of exchange, that means how import of plant genetic resources into the country is governed by the Plant Quarantine Order 2003, and there are only two requirements. One is the import permit, which is issued by the Director NBPGR, and here I am talking of only import for research purposes. So under this PQ order, there are two channels for import. One is for research and the other is for commercial purposes, that means bulk imports. Those import permits are issued by the Department of Plant Protection and Quarantine. Director NBPGR is authorized to issue permits only under PQ order for research use. And then the second requirement for import is the phytosanitary certificate issued by the exporting country. And based on the import permits, there are, if certain additional declarations are required by the exporting countries, those have to be included in the phytosanitary certificate based on the import permits. Then for export of genetic resources from the country, I have listed above the four type of material. One is the Annex 1 crops of the ITPGRFA, for which the permission is required from Department of Agriculture, Research and Cooperation. And then for exchange of such material, the standard material transfer agreement of the treaty is used for such transfer. And second category is export under collaborative research projects. Here we sign the Indian MTA and the concerned departments, they give approval for export. And then for basic and emerging research and students going out of the country, permission from NBA is required in Form B. And if the export does not belong to any of these categories, then the permission of National Biodiversity Authority is required as per the BDA 2002 and Rules 2004. Then collection of resources from India for commercial utilization. This is covered by Section 7 of the Biodiversity Act. Here, if you want to commercially utilize the material, then you have to inform the NBA. Then if you require any genetic resource within the country, then NBPGR gives you service for that, and that we require you to sign the material transfer agreement and also request you to maintain the IC and EC numbers for such resources. So this is the last of the slides. And basically, I would like you to have interaction on these issues where we are not clear on any of these issues. We can discuss and then take on some of the questions. Thank you very much.
Thank you, ma'am, for giving very exhaustive, and I hope all of the participants must be benefited with this lecture. And I understand that this is very new area for all of you, I think, but really it is a need of our, because we have so many PGR activities and it is continuously increasing day by day. So we have to also look the policy aspects. So ma'am, if you will permit, then we will take some questions.
Yes, sure.
Okay.
Basically,
And,
I wanted this
just
to
I
be
am
an
seeing the chat
interactive.
box. Dr. Vinod Kumar from JNKVV Jabalpur. He asked that he is asking that how to use material which is having no information regarding source and pedigree. So what can be done?
I didn't understand your question.
Please unmute yourself. Please unmute yourself.
Hello, ma'am. Good afternoon.
Ma'am, please unmute yourself. Your voice is not coming.
Okay, yeah, tell me.
Good afternoon, ma'am. Myself, Dr. Vinod Kumar from Pawar Kheda, JNKVV. My question is that if any material we have, but their source is unknown, what from collection and if pedigree, then why we can use the in our breeding program.
Yes, it
How
is.
we can use the in our breeding program.
Yes, it is not advisable to use such a material. Or you can do one more thing, that you can characterize that material,
Yes.
generate a data,
Yes, yes.
and then
Yes,
you
ma'am.
get IC numbers from NVPGR. Okay.
Yes, yes.
Where you can basically
Thank you, ma'am.
define from what institute or what place you are working, and you generate that set of data so that you
Yes,
know
yes, ma'am.
it is, but you don't know the identity. So we can give you another identity so that you can be sure of what are the characters of that material. You get an
Yes,
IC numbers and then use that identity for further breeding program.
Thank you, thank you, ma'am.
That is my suggestion. Yes.
And second question from Dr. Vikas Pawar. Why India is not member of UPOV?
Actually, UPOV is basically for the plant breeders' rights. There are very strict. So our Indian Act has a very strong component of farmers' rights. That means we have to give farmers the right to reuse the material, sow the material, exchange the material, which is not as per international standards, where they say that if you give farmers so much right, the commercial benefit which the breeder gets will not be possible. So the rights of the breeders are diluted by the farmers' rights. But since we are a country where own system of plant variety registration, so although we are compliant to WTO TRIPS, we are not members of UPOV, and we don't feel the need also. So it is not necessary that we should become or we should not become members of UPOV. Okay.
Hello. And one more question from Dr. Lalu Prasad Yadav. Is there any policy or act for the planting material introduction from other countries by private sector?
See, for research use, they have to come through NVPGR. And if they want to import planting material in bulk, they should go to Department of Plant Protection, Quarantine and Storage and get import permit from there. And then the material is imported and examined by the DPPQ authorities and then released. So private sector, public sector rules are just the same.
I hope you got your right. You got the answer, Dr. Lalu Prasad. Okay. Is there any question? Anybody else is willing to ask something?
Madam, I am Dr. Tarak Nath Shah from Directorate of Floriculture Research. I have a query.
Yes.
Am I audible, madam?
Yeah, a little. Can you speak little louder?
I am Dr. Tarak Nath Shah from Directorate of Floriculture.
Mm-hmm. Yes. What is your question?
The question is that, madam, in India, we are having lot many tuberose varieties, around 20 to 23 tuberose varieties, but all are white in color. Tuberose.
I didn't get you.
In India, we are having 23 tuberose varieties. Tuberose, Rajnigandha.
Tuberose, okay, yes.
Rajnigandha. So all are white in color. Right, madam? But the thing is like that in other countries, like in Mexico or Taiwan, they are having pink colored, yellow colored tuberose. Right. So, but I do not know what is the status of protection in India or other countries. They have entered India.
that varieties are available with the private nurseries. Can we use that colored tuberose varieties in our breeding program?
Those material, if it is already available in the private nurseries and it is available to farmers for cultivation,
Yes.
then your breeding programs, it is illegal. You cannot do it. So you must import it first legally from those countries, maybe for research, maybe for commercial. Then only you can exploit those for developing or breeding in your programs. So because in our country, if you want to protect those resources, protect the newly developed varieties, you should have the full parentage of that variety from where you have taken the parental material. Or if you want to import and commercially sense of commercially exploiting those varieties in India.
Madam, your voice is cracking, madam. You are not audible, madam. Your voice is cracking. Yes, madam. Please.
Okay. If you are talking of tuberose, there are so many other crops in which we have illegally introduced varieties, or the nursery have got it from somewhere and they don't disclose the source. In all floriculture or horticulture crops.
Actually, this is actually, madam, vegetative.
As a responsible scientist, you should
no,
not.
No, obviously, madam. Actually, it is a vegetatively propagated material, so
Yes.
it is easily proliferated like that way. And madam, we have written to NVPGR also. NVPGR has also said like that way. You directly contact with the concerned means breeder or the country. But that when we communicated with the same thing with that country or that breeder or that nursery, they are simply refusing that due to their legal problem. They are not supplying it to India.
Because yes, because if their variety is protected in that country,
right,
they will use the commercial benefit of that. If they you should talk to them for licensing them to you with and paying them certain royalties. Only then you can use it.
Okay, madam. Okay, madam. Right. But after 15 or 18 years, we can use.
After 15, yes, if it is available and you have the record. See, there also the record is important.
Yes, madam. Yes, madam. Yeah. Right. Okay, thank you so much, madam.
And we will take just last question, I think, from G. Thanavindan. For underutilized fruit, have any specific policy to transfer from other countries?
No. I said all the genetic resources of all crops, all, even plant, animal, fishes, the rules are just the same. There is no distinction when we talk of genetic resources. Only thing I talked was what is the difference between PGRFA and general biodiversity. The rules are just the same for exchanging. There is no difference. So if you want to import or export underutilized species, rules are just the same what I have listed.
And also Dr. Vikas Pawar has asked one more question, ma'am. We will end here because he asked that.
Okay.
He asked that because there are a lot of questions. I understand because this is new area for everyone. Na? So everybody is very curious to know these things. And mostly the people are getting very difficult to exchange the material, That's why they are curious to know. That's why they are.
And also we have a separate lecture from Dr. Vandana on exchange procedures. And there is a practical also. But yes. You can ask any questions I would like to answer.
One more we will take from Dr. Vikas Pawar. He is asking that can we use material for research purpose under researcher exemption?
Yes, you can. Yes, you can. Okay. Only two conditions are listed in PPVFRA. One is that if you want to use the material continuously for as a parent for every generation, then you have to take permission from the breeder. or if you are using it for developing an ED, that is essentially derived varieties, you want to develop a GMO or any genetically modified variety, only then you take the permission of the breeder. For other normal breeding processes, you don't need the permission of the breeder. Researchers are exempted from using the variety.
Okay, so thank you, madam. You have addressed all the queries, and you have addressed almost more than 10 questions, I think. And thank you for sparing your time and giving your thoughts and opinions. Definitely all participants will be benefited with this thing, and they will be, and I would like to assure all participants, if you are facing any difficulty, you can directly write to exchange head or respective resource person's email ID. You can mention your queries, and they all will be responding your queries. So thank you, madam, and for your time in this thing. Thank you. And we will rejoin once our next resource person, Dr. Badal Singh and Dr. J. Arvind will join.
Thank you very much, Kuldeep.
Thank you.
तो कंट्रोल हो जाएगा।
Badal, Dr. Badal, are you
तो
around?
इसलिए हम डालने हैं।
Okay.
I'm here, sir.
So,
यही वजह से इसको डालने हैं।
Dr. Somir Singh, please mute yourself.
Sir, let me call Dr. Arvind sir to get online.
Please do it. No problem. So actually, in-person training, we are regularly organizing, visit to field gene bank, national gene bank. But of course, I hope you understand it is very difficult to arrange field visit. So we have tried to arrange virtual visit for you. So we have Dr. Badal Singh and Dr. J. Arvind. They are from Division of Germplasm Conservation. They are looking after the oilseed crops in National Gene Bank at ICR and NBPZR New Delhi. So they will arrange your virtual visit to National Gene Bank, and in the due course, we will be having also detailed lecture from the gene bank perspective with different crops, different crop groups, and I hope you all have seen the schedule also. So I don't think there will be, you will have enough opportunity to know each and everything about NBPGR in different aspects, let's say from collection, conservation, evaluation, utilization, genomic resources. So Dr. Badal, can you start?
Actually, we have arranged our material into two parts. The Dr. J. Arvind will elaborate on the basic technical parts of the gene bank system and the seed conservation system. And after that, the virtual tour will be handled by myself.
So whether he is online or not?
I have called him, and he will join soon.
Okay, so let him come. We'll wait for that. No problem. And for all participants, this is my general suggestion. Please keep your rename yourself in the Zoom account like somebody is coming with iPhone name. So it will be very difficult to identify them. So please keep name as we have sent you in the WhatsApp group, or Mr. Vijay Mandal also informed you in the morning how to rename yourself. So please do that. This will be difficult for us. Otherwise, we are unable to identify yourself. So please keep name and along with the institution name. So Dr. Arvind is here, and Dr. Arvind is a scientist in Division of Germplasm Conservation, ICR and NBPZR. He is looking after the oilseed crops along with Dr. Badal Singh. Recently, he has done his PhD from IPK Germany on barley and other cereal crops. He has standardized and some calibration models on germination or some, and he has also expertise in developing some, tools also based on the R software. So Dr. Arvind, it's over to you, please. Dr. Please unmute yourself. Unmute yourself.
Okay. Oh, okay. Oh, I hope now you can hear me.
Yes.
Yes, I'll share my screen.
Sure.
Yes.
No, it's coming. No issues.
Yes. Okay. Good afternoon, everyone. I am, as mentioned, I'm Arvind, and today, along with my colleague Dr. Badal, we'll be taking you on a virtual visit of the Indian National Gene Bank. So initially, I'll be giving a brief explanation, outline of the kind of work we do in maintaining the material, seed material in the Indian National Gene Bank. And later I'll hand it over to Dr. Badal, who will be taking you on a virtual walkthrough of the gene bank. So the present facility of the gene bank was established in 1986 when the first the cold storage modules were established in NPPGR. And the mandate is for the ex-situ, that is the conservation of material away from their origin for long-term conservation. So basically, we are maintaining the long-term base collection that is conserving the material for a
Dr.
long period
Somveer
of
Singh,
time.
please
30
mute yourself. Sorry. Dr. Somveer Singh, please mute
Yes,
yourself.
it's all right.
Because please.
Yes, as I was telling, long-term conservation means conserving the material under low temperature of -18 degrees Celsius for 30 or 50 years or more. And most of the collections are held in primarily in the seed gene bank because most of the crop plants have seeds of orthodox nature, that is, which can be conserved for long periods of time and by reducing the moisture and temperature of storage. Also, the vegetative propagules, cell cultures, embryos, everything are kept in vitro and cryogene bank. And this makes the Indian National Gene Bank the second largest gene bank in the world. And in NBPGR, there are different, the responsibilities are shared by different divisions and units. And all these activities revolve around the gene bank. As I mentioned, the present long-term storage module in the present form was established in 1996, but before that in 83, the first cold storage module, but it was for 10 degree and 4 degree Celsius was established with 30,000 capacity. And later for -20 degree cold storage vaults were established. And the large scale long-term modules, 12 modules were established and under the Indo-USAID program in 1996. And this year, it will be the 25th year of the establishment. And in 2021, all these modules were upgraded to a more efficient modules. And from these modest beginnings, now the collections in the gene bank have grown to over 463,000 accessions, and they belong to 1,700 species. And the major collections include cereals, and 1/4 of the gene bank collection is rice. And other major collections include wheat. Then we have oilseed brassica, groundnuts, sorghum, then chickpea. So these are the major collections which are held in the gene bank, and these are also the major assemblages of this crop in the world also. These collections also include, they comprise of around 95,000 land races, around 25,000 elite lines, 12,000 improved cultivars, and also 8,500 genetic stocks. And we also have concise and specialized collections which capture the diversity in these large collections. Such as core and mini core sets in rice, wheat, barley, brinjal, and recently in mustard, then safflower, sunflower, chickpea, mung bean, PGNP, and recently also in lentil and cowpea. And the conservation strategy for the seed gene bank, it's based on a series of mathematical models, which is known as seed viability models, which shall tell the relationship between seed viability, the temperature and moisture content of storage, as well as the storage period. So we can predict based on these models for how long we can store a particular species under particular storage condition. So the factors that affect the survival of seed under storage condition, it is essentially the temperature, the moisture content, as well as the initial seed quality. The temperature and moisture content we can regulate it through control it through our equipments. But for the initial seed quality, for that we have to process the material so that it meets. We can ensure that they meet the initial seed the quality requirements. So this forms a core activity in the gene pack, and I will just briefly tell what are these activities, because in other lectures I'm sure that my colleagues will be explaining all these aspects in detail. So once we get a material in the gene bank, we have to see whether all the supporting documentation is available, and it differs based on, for example, varieties. The documentation is different. For exotic material, it's different, and for indigenous material, the kind of passport data it is there or not. That information should be there, and then we need to see whether it is whether it's already present in the gene bank or not, to avoid any sort of duplication. And then we need to ensure that the seed quantity is adequate. So the seed quantity is determined based on population genetic models, which ensures that any accession what we're conserving, these are not pure material, these are populations. So we need to also able to conserve the within accession diversity. So based on the nature of pollination and the population structure, the amount of rare alleles that can be conserved in the minimum number of the population size. So it's 2000 for self-pollinated crops, 4000 for cross-pollinated crops, and for wild species it's relaxed to 500. And then whether the seeds are clean and whether the seeds are pest-free. The pest-free conservation is ensured by our colleagues in the plant quarantine division. Then the seed material is dried initially through walk-in seed dryers, which can reduce the seed moisture content to around 7 to 8%, and then further drying is through machine dryers. Badal will be showing you machine dryers later on, and finally this moisture content is reduced to around 3 to 7% moisture content. And this is monitored and estimated through the ISTA standard methods. Then the viability is tested. So to ensure that we are at the starting point itself, we are having a good seed viability, and the threshold is 85% for most of the crops. But for some wild species, medicinal species, forage species, etc., it's relaxed to around 50 to 70%. And finally, the seeds are dried using vacuum sealing in a tri-layered aluminum foil packages, and these are really tough packages which can which can take a lot of abuse and won't break, and can maintain the moisture content under low temperature conditions. Because in under minus 18 degrees Celsius, we cannot control the moisture, we can only control the temperature. So in that conditions, we have to use this kind of vacuum sealed packages. And then each material is documented in the in our in-house database. And each sample is given a unique national identity. It is either IC or EC number. IC indicates the indigenous collection and EC indicates the exotic collection. And so that we can keep track of where we have stored the material, how much sample is left, and what is the moisture content and viability, and where when we have to take it out for monitoring. And once we keep the material in the zinc bank, we just don't forget about it. We also retrieve the material at regular intervals, at 10 or 5 years intervals, and then they are monitored for their viability. Also, whether the seed quantity is reduced because of supply. And then if a threshold level is breached, that is, for example, 85% of the initial seed viability, then the material is subjected to regeneration. And regeneration is essentially a population bottleneck process, so we try to avoid it as far as possible. But only if the threshold is breached, then we go for regeneration, and it is done through the network of our regional stations, headquarters of NPPGR, as well as the national active germplasm sites, which are all spread all over the country. And for some wild species such as wheat wild species and all, we go for special conditions also for regeneration. For example, here a long photoperiod condition for wheat. Okay, now I'll stop sharing my screen and hand it over to Dr. Badal, who will be taking on a virtual walkthrough of the gene bank along with Anjali.
Okay, Dr. Saab, you may have to, maybe you have to unshare your.
Yes, I'm doing it. Okay, I've unshared my screen. Can you go ahead?
Okay, I will go through this virtual walkthrough. I'm sharing my my video. Can you see this video, everyone?
Yes,
Okay, so let's have a virtual walkthrough to the one of the unique facility presented in the country. It is actually a national heritage. So we are dealing with preserving the vast majority of the diversity present in our crop genetic resource. So let's walk through this. So it's upgraded system. It's our corridor through we go to the system. Recently it has been upgraded and has been inaugurated by our Honorable Minister, Agriculture Minister, Sri Narendra Singh Tomar ji. And so we will move to these modules, the vaults. First, this is the walk-in seed range chamber, as you have been heard by Dr. Arvind sir. It's an facility which has provided the drying system, which maintain temperature is 15 to 20 degrees Celsius, and the 15% RH. So you know that when we have the low RH, then the seed drying process start. It's the walking drying chamber. It's the seeds are kept in the basket, and then we have got 12 modules. It's the outer side view of the module. We have got 12 module, one MTS in this new building and 4 in old building. So it's the and we used to maintain in every module we have maintain the different crops like this. This is for the pseudo cereals. We have kept all this listed, and so we go inside this vault. We can see that the seeds have been arranged in the basket, and these cells are numbered. The baskets are marked, these seed packets are barcoded. In the upgraded version, it's barcoded. Earlier, it was not barcoded. You can see how we kept these packets. And we also provide the location likewise. This is the racks, A, B, C, D racks, and cells. Basket number. So according to this, we arranged all the seeds here. So it's a machine drying system. It's also work on the same it's a 15 degrees Celsius and 15% RH and 15 to 20 degrees Celsius. It's a machine drying system, and here we are going to the packaging system. It's a tri-layered aluminum package, and we have to do the vacuum packaging to remove any excess moisture present in the packet. It's very critical. It's an important step while packing, that any moisture or air should not be remain inside the packet. So it's a machine drying system to remove the excess air. Vacuum drying system. Actually it's a vacuum drying vacuum packaging system, and after that, this is the monitoring system. Here we got all the logs and stats of temperature, moisture, temperature fluctuations, suction pressure of gas, the discharge pressure of gas. And we are walking through this, the units. These are the 2 units are present per module to get the 24/7 compression facility. And it has been set for the 300 minutes, and automatically it will shift to the second unit for the backup. And this is the central management system. We remotely can access all the parameters here, like moisture, temperature, and suction pressure, all this. And there is the alarm system. The allows, if the one of the unit is not working, then meanwhile this automatically this is shift to the next backup unit, and in meanwhile we have to make it correct or see the problem, what is the reason. There will be a yellow alarm, red alarm.
Hello.
Badal?
Hello, Dr. Badal. Hello.
Yes, sir.
आपकी आवाज नहीं आ रही है.
हाँ sir.
चलो अभी
Sir, can you hear me?
Now it's okay. So thank you, Dr. Arvind and Dr. Badal Singh, for taking extra efforts for arranging virtual visit. Indeed, it is very difficult. If we are doing in-person training, it will be very easy for us. You have to show, and you will also cherish the moment by seeing the National Gene Bank. But Dr. Badal Singh and Dr. Arvind has taken a lot of efforts for making this, presentation and virtual visit, of course. So I request any of the participants, if they are willing to ask any questions regarding the gene bank and how they are working in, because they both are responsible for the oilseed germplasm. Those who are working for oilseed germplasm, they may ask any question regarding, from gene bank perspective. So floor is yours. If anyone would like to ask questions, they may ask through chat box, or they can directly ask by raising their own hand. Anybody?
Sir, we have to answer chat box or
somebody? No, let me see. If they are asking, then I will respond.
There is a question in the chat.
Okay. And Dr. Amit, you can reply after seeing chat box also. No problem.
Yes, the temperature is minus 18 degrees Celsius, and the moisture content, it varies. For example, for cereals and all, it is 6 to 7%, but for oil seeds, it can we can bring it down even further. How low we can store oil seeds, oils?
So theoretically, we can store in the long term. We can store for the longer time, but yes, there is a difficulty in oilseed because its storability is not quite good. So we have to take additional care for that. Regarding we have to shorten the monitoring duration if its availability down, and yes, but I have got only 1 year experience. Dr. Arvind may say.
If I can add to that, it is crop specific. Most of the oil seeds are really good storers, except for soybean, where there is a lot of interspecific variability. Within species also, there is between accessions also there is quite a lot of variability. But otherwise, because of the oil content, they are generally good storers.
For oilseeds, it require high moisture. I don't think for oilseed it's high moisture content is required. Actually, with comparison to starch content seed, oilseed lose moisture more rapidly, and it can be stored at 4 to 5% moisture content. How to maintain 0% of all the seed crop and how to do. You may
How to
answer one of the.
Yeah, I'm trying to figure that out. How to maintain germination percentage for all the oilseed crops and how period of time can. Yeah, basically it is we have to control the temperature, the three factors, that is the temperature, the moisture, and the initial seed quality. If we can have that, to ensure that we can have a good there's a question for cryo. I think that there will be lectures in detail for cryopreservation. Isn't it right, Kuldeep?
Hello. This is we have also separate training lecture on cryoconservation also. So realizing the we have to ask question which is that I already told you. You have to ask question directly relevant to the concerned topic, because we have shared you the schedule also, so you can ask these questions in respective presentations of the resource person. This is a very good opportunity because these two are the only key person who is representing the oilseed crop in NBPGR. So this is good opportunity. Those who are working in oilseed crops, they may ask questions directly.
Yes, and coconut and oil palm, they are not orthodox species. So
actually, this seed bank system is only for the orthodox species. I think, sir, there is no class. This is our first class regarding PGR, conservation aspect.
This is first class.
So that's why they didn't know that actually
there will be separate lecture on the seed storage behavior, whether it is orthodox and recalcitrant seeds. So you may be enlightened during the lecture which has been taken by a specialized person. Those who are taking the practical and lectures also will be there. So these specific answers will be given in that respective lectures. You can ask anything regarding oilseed crops, specifically. Oilseed means orthodox oilseed crops.
Actually, I think one general thing I would like to say that every species or every oilseed species have a different seed biology storability behavior, and we have to work out on that. For if we get any oil rich species, first we have to get to work out their storability. And based on our experience, like we can store groundnut. If someone is asking groundnut MTS. You should know the MTS we maintain in a medium term storage. The temperature is 4 degrees Celsius, and RH is 30 to 35% moisture for medium term storage. For long term storage, it's minus 18 degrees Celsius. Yes, we can store groundnut in medium term storage. Based on we have to work out and there will be some genotype also, variability also there. So we have to work out their storage behavior in relation to the time. If you have got any different oilseed species other than this groundnut or mustard or anything. First, we have to develop a particular protocol for this conservation.
Anybody else? If not, then we will thank our resource person, Dr. J. Arvind and Dr. Badal Singh, for taking a lot of efforts in preparing virtual visit to National Gene Bank. And if for any resource, for any lecture, if I request all the participants, if they are willing to ask any question, they may ask directly, or they may also after the training also, anytime you can contact this person directly and they will respond you. So thank you, Dr. Badal Singh and Arvind.
So
Thank you for sparing your time and giving your thoughts and opinions on the lines. And I would like to I'd like to say for the participants, we would like to break here, and we will be sending day-wise schedule regularly through our WhatsApp group. And please be on time and switch on your videos during the initial for our screenshots and attendance marking. And anything if you'd like to ask general question for regarding training and how it will be going through for 21 days, let me know. If you want anything regarding training, organization and anything, do let me know. If you want any clarification for training as per the organizing perspective. If you want any clarification regarding how we are going ahead. And one more request, if somebody, because we have arranged some lectures from international institution, they are from CGIAR center and recognized person from a specialized field. So it indeed it's not good to, if you are using through mobile and we are roaming around, we are traveling through. So please try to avoid. It is better if you are, maybe you are busy at the moment. So please switch off your video if you are using through mobile, because it doesn't look good and it is also distracting the resource person also. And we'll be sharing all our training compendium in the mid, either before the voluntary session. We'll be sharing the training compendium also in the form of e-publication, and all relevant pro forma and forms for sharing material, registration of the journal plasm, taking IC number, everything, every pro forma guidelines, whatever has been developed by ICR and BPCR, we will share you at the end. Or also we have arranged lectures, practicals similarly based on your needs also. It's not like that we have arranged lectures which has been isolated. It is all well connected with each other. So based on your need, like since collection, how we are going for collection mission, everything, all the heads of the division will be taking lectures. For any queries, you can directly ask and this thing. For me, if you want to ask anything, then I'll be happy to answer it regarding organization perspective. And beside this, I am working on pulses. So, okay. Anything? Otherwise, we will close it.
Sir.
All the.
डॉक्टर साहब क्या सबकी एक बार फोटो लेनी है या वीडियो?
हाँ प्लीज मंडल जी एक बार ले लीजिए.
एक बार सबसे रिक्वेस्ट कीजिए कि वीडियो ऑन करें अपना.
I request everyone, please switch on your video. Switch on your video, then we will take one screenshot, because in some screenshots everybody is not represented. So I request each and everyone, please switch on your video. Dr. Somveer Singh, Dr. Vinod Kumar, okay, done. And who else? Everybody. Dr. Sujata from Piha, okay, done. And Dr. JM, Dr. TK. Please, I request everyone, please switch on your video. This will be the first pick with all videos and everyone. Dr. Reena Mehra is laughing too much. Okay.
Wait. Okay, thank you very much.
Thank you one and all, and we'll close for here. Thank you Mandalji for your last remark. Thank you. Okay, bye.
सर मीटिंग क्लोज कर दूं?